so I think everyone agrees that brain is a machine that processes information and make memory and retrieve it when needed. And as a modern human people, our our major reference for machines that process information are computer, which gives sometimes like metaphor comparison like this one, which I think are confusing. And why do I think it's confusing? Because the way that computers have been designed by man in a world where energy was plentiful and not limited, is that basically computers have codes that they can execute, and they will execute them on demand, independently of whether energy is available and the quantity of energy available. So of course, if there is no battery or if the power is shut down, then they will not be able to work anymore. And but otherwise, if energy is available, then they will do what they have to do independently of what happens. And I don't think this is how the brain really works, but unfortunately, this is the way in neurobiology many people tend to study the brain, meaning that unless they are interested in a specific pathological context where energy defects can occur, so if they are working on healthy, young brain, well-fed, they will basically consider that energy is there and the brain takes energy when it needs, and then it does what it has to do. But contrary to computers, brains and nervous systems in general have been shaped by evolution over millions of years in contexts where energy was not always available, and most often nutrient availability was a limiting factor. And this is, I think, the reason why, contrary to this maybe too simplistic view, actually really understanding how brain function needs to take into account the fact that energy metabolism, and when I say energy metabolism, I'm not only talking about the final output, which is ATP production, but also the type of metabolic pathways that are mobilized and the other outputs, chemical outputs than ATP that they can produce. This whole energy metabolism really is intricated with brain function and can shape its outcome. And in our case, the outcome that we are interested in is memory, and we study memory in Drosophila. So you may wonder why we use Drosophila as a model organism. So it's not a course on Drosophila neurobiology, so I will just go quickly on this. But the main reason are that it's quite simple. There are only 100,000 neurons in the Drosophila brain. So much less, of course, than in a human or even any mammalian brain. But still, they are able, as we will show later, this brain is able to encode quite a sophisticated set of different memories with very interesting memory dynamics. And because of this numerical simplicity, over the past decades, the community has reached a description of the neural circuits underlying memory formation and retrieval down to single cell resolution, which is then now very convenient to ask very specific questions about the mechanism underlying the regulation of memory. Initially, Drosophila was used at the beginning of the 20th century to unveil the rules of genetics. So the genetics of in this species is very well mastered, and then it has become very easy to do transgenic animals, and we have what we call a genetic tractability that allows manipulating any genes, and in particular genes involved in metabolic pathways, only through genetic manners, meaning non-invasively, and being able to target specific cells and, that we want to study. And in addition, and this is more the specificity of our lab, we have developed over the past years methods using some fluorescent metabolic sensors, like the one that Nathalie has talked about, but not only, that we will use for in vivo imaging to monitor in vivo those energy fluxes with a cellular resolution. I will now give you an overview of our more or less recent works. I will start first by giving you a description of the Drosophila brain and the circuits that are involved in memory encoding. But I think I will also try, beyond the data, and maybe the detailed mechanism that I will show are not of interest for everyone here, but I will try to extract some messages about how energy shapes the memory encoding and the brain function that could be of interest. So, the protocol that we use in the lab is a protocol of associative memory, in which flies are trained to associate one odor with electric shocks, so a negative stimulus. So this is a Pavlovian-like protocol. They will smell sequentially two odors, the first one of which will be associated with electric shocks. And so this electric shock, electric shock associated odor, which is called the CS+, will become predictive of something negative, and then the flies will learn to avoid this odor. So just to make things a bit more concrete, this is how we do in practice. We will we train group of flies, and we will always measure memory of groups of flies, not individual flies. A group of flies will be put in a machine that we call a barrel, and so 50 flies will be put in this cylindrical chamber, which is covered with an electric grid that is connected to a voltage generator. And each barrel is connected to an airflow system, so that the computer will control the delivery of one odor that will flow through the barrel and the delivery or not of electric shock. So the conditioning is fully automatized, and we can parallel this to have a high throughput of fly conditioning in parallel. After conditioning, we will test the memory of each group of flies that have been conditioned, and this we do in a machine like this, which we call a T-maze, inverted T-maze actually. The group of conditioned flies will be put in this elevator, and then the elevator will be put down, and they will have the choice between two tubes where the two odors that were used during the conditioning are arriving, so the fly can choose which compartment they want to go. Normally I just have a small movie here. So this is how it can. You can see here, for example, that from here the fly mostly go to the right and avoid the compartment of the odor A where the shock had been delivered. So they are given 1 minute to choose, and after 1 minute we collect them, and we count the number of flies in each compartment, and from those numbers we calculate a memory score. Basically, if there is the same number of flies in both compartments, it means that there is no memory, the score will be 0. If they all go to the compartment avoiding the electric shock, the score will be 100. And so the seminal studies that this protocol have been used for decades now, and the seminal studies have looked at the persistence of the memory or the efficiency of this protocol. So you can see here that immediately after learning, the memory score is actually very high. So it is an efficient way to teach flies to avoid an odorant. And the memory will decay over the course of a few hours, and after 1 day, it's almost completely gone. So how does it work? How is this encoded in the brain? So this is magnification of a fly, a Drosophila head. in blue here you have the brain, and here a structure that I will talk about a lot, which is called the mushroom body. And the mushroom body is in the fly, but also in all arthropod species, the central structure involved in associative memory processes, especially olfactory memory. Why is it so? First of all, of course, because it's connected to the olfactory processing pathway. When a fly smells an odorant, chemoreceptors are activated here in the antenna and here in the maxillary palps. And sensory neurons, which carry those chemoreceptors, project on a first structure that you see here, which are called the antennal lobe, where basically the projection of the neurons are segregated according to the nature of the odorant. And then from these antennal lobes, projection neurons will send their axons into the brain and connect two regions. One, I will start with this one, one on the side here, which is called the lateral horn, which is more involved in the innate odor behavior. And so the famous mushroom bodies, which will be responsible for the adaptive and learning behavior. So let me describe those mushroom bodies in more detail. So they are made of, so it's a bilateral structure, as you can see here, symmetrical, with roughly 2,000 neurons per brain hemisphere. The cell bodies of those neurons, which you don't see here, would be at the back of the brain. And each neuron would have a dendritic part in this region, which is called the calyx. So the calyx is the place where neurons will receive the olfactory information. So this is basically the input site of mushroom body neurons. And mushroom body neurons will then send their axon through the brain here and ramify. So we can define, but it's not really important for the talk here, 3 different subcategories of Kenyan cells. But what you need to understand is that here, axon can ramify and form different lobes. And as we will see later, these axonal sites are the output region of mushroom body neurons. In addition to neurons, there is also non-neuronal cells in the brain, and as in the Drosophila brain, and like in almost all other brains. So these non-neuronal cells are called glia. 5 different types of glial cells have been described in Drosophila. The most important for us today is this green one here, which is called cortex glia. And it's a specific glial cell type which is exclusively in contact with the cell bodies of neurons, so that each individual neuron will be enwrapped by cortex glia, and a single cell of cortex glia will connect roughly a dozen neurons. But all in all, on all the brain, every neuron is connected to cortex glia. So how is memory encoded in this system? So I told you that the lobes of the mushroom body neurons, so this part here, are the output sites of those neurons. And actually, on these sites, mushroom body are connected to a set of what are called mushroom body output neurons, which are organized in a very specific way. You can see that so there are 15 different cell types, but each cell type will be connected to a very specific and mutually exclusive region of the mushroom body lobe, thus defining somehow independent compartments that can process the output of mushroom body neurons. And what is interesting so what I show here is not only our lab work. This first introductory part is, of course, the result of the work of the learning and memory Drosophila community for more than 15 years. And in particular, those movies are from this reference. But what's a striking result that the community has reached as a whole is that, functionally speaking, those mushroom body output neurons can be divided into 2 parts. Ones that will mediate an avoidance behavior, and this corresponds essentially to the one that will innervate the medial lobe, here in orange. And another one that, when activated, will drive an approach behavior. The one indicated in blue here that will mostly innervate the vertical lobes of the mushroom body. And in a naive animal that has not been trained, if the animal smells an odorant, then the mushroom body neurons will be activated, and they will equally activate basically the whole network of mushroom body output neurons, so that the output of the mushroom body is a balanced drive between avoidance and approach, so basically neutral behavior. So how is olfactory learning implemented on this system? It is implemented through a second class of neurons that innervate the mushroom body lobe, which are dopaminergic neurons, and those are input neurons. And you can see that they are also organized very similarly to the mushroom body output neurons, so that each type of dopaminergic neuron will correspond to one type of mushroom body output neuron here. And when we do a conditioning, or when the fly receives either a positive sugar, for example, or a negative electric shock stimulation, then those dopaminergic neurons will be activated also in a very segregated manner. Punishing or negative stimulation will mostly activate dopaminergic neurons on the vertical lobes, and positive stimuli will activate neurons on the medial lobe. And so the way to go quickly to the principle of memory encoding, the way memory is encoded is that during learning, when mushroom body neurons are activated by an odorant, and simultaneously dopaminergic neurons are activated by electric shock, the coincidence between both dopamine signaling and mushroom body neuron activation will induce a local modulation of the synapses between mushroom body neurons and the output neurons. And this local modulation, and the synaptic rule is a depression of the synapses induced by dopamine, will induce both a decreased drive in the approach network and an increased drive in the avoidance network. So that when the fly later on smells deodorant again, then instead of a neutral output of the mushroom body, then the sum of a decreased approach and an increased avoidance will induce an avoidance behavior of the animal. Okay, so this is how memory is encoded and what happens during the retrieval of memory. But in this first part, I did what I criticized at the beginning. I talked only about information processing, about the logic of encoding, but without caring at all about the energetic part behind this. So this is now what I will start on, coming more to our work. So what happens in terms of energy metabolism when we train flies and when they form their memory? So, brain energy metabolism is a highly controversial field with a lot of debates about which cell types does what and takes which metabolite, sorry. But there are two things that make a consensus. The first one is that in the regular condition, the brain essentially, if not exclusively, relies on glucose, contrary to other organs that can use fatty acid and so on. And that neurons strongly rely on oxidative metabolism in mitochondria. So on this slide, which is very complicated, I just put a general scheme of glucose metabolism to remind you of a few important points. Not everything on this slide is important. What is important is that, so this is a scheme of complete glucose oxidation through to ATP production, which can occur in any cell. Glucose enters the cell and is then first partially oxidized through this sequence of reaction, which is called glycolysis. and the output of glycolysis, which occur in the cytosol of the cell, which will produce pyruvate. This pyruvate can be converted to lactate or to other metabolites, as we will see. Or it's also the primary metabolite that will be consumed by mitochondria for oxygen-dependent energy production. Pyruvate enters mitochondria, and I emphasize this enzyme because it will be important later on in the talk as well, is converted into acetyl-CoA by an enzymatic complex which is called the pyruvate dehydrogenase. And that acetyl-CoA is processed by the famous Krebs cycle to produce reducing equivalents. Those reducing equivalents are consumed by the mitochondrial respiratory chain to produce ATP from ADP in the end and consume oxygen. So the important point here is that pyruvate, in terms of glucose metabolism, pyruvate here is both important for mitochondria energy production, but also it's also the output of glycolysis, which is the other ATP producing pathway. So, and we had the chance that a couple years ago, a group in Chile engineered a fluorescent genetically encoded sensor to monitor the concentration of pyruvate in cells. So it's a Fred sensor, meaning that two different fluorophores, one blue and one yellow, are on both sides of a peptidergic of a peptide of a polypeptide that can bind pyruvate. And upon pyruvate binding, the conformational change that will occur in this peptide will change the distance between the two fluorophores so that energy transfer from the high energy one to the low energy one will be modified. So if we excite the blue fluorophore and monitor the fluorescence from both the blue and the yellow, and we do a ratio of those two fluorescence emission, we get a signal that is directly correlated with the intracellular pyruvate concentration. And in practice, to do that, we do that in vivo in the Drosophila brain. So under a 2-photon microscope, we can glue a fly, do a small surgery to have access, an optical access to the brain. And you can see here pictures of mushroom bodies of this fly expressing the pyronic sensor with, as expected, emission in both the blue and the yellow channel. Now, expressing the sensor is good, but it's not sufficient. When we want to measure metabolic flux, in the end, the concentration is not the important parameter. What is important is the rate at which pyruvate is produced or consumed. And actually, here on this baseline, you can see that if you just open the head and look at the pyronic signal, you don't see much change. So it's the same principle as what Nathalie has showed in her talk before. What we did is we applied a pharmacological treatment to block the flux of pyruvate and see pyruvate accumulation to have access not only to the pyruvate concentration but to the rate of pyruvate consumption. The drug that we use is sodium azide, which is very convenient because it's water soluble, so it penetrates very well into the brain and enriches the neurons. Sodium azide is a blocker of the mitochondrial respiratory chain, so that when it's applied, mitochondria stops consuming pyruvate, and so the pyruvate, instead of being consumed, accumulate in the cell. And we could show that the slope of pyruvate accumulation, as we can measure here, is directly correlated to the activity of the pyruvate dehydrogenase complex that I mentioned before. So with this protocol, which is in the end quite simple, we have a very convenient access to the cellular pyruvate flux in mushroom body neurons. And so what we did is we did these experiments after an olfactory conditioning. So we conditioned the flies with the idea of measuring, of looking at the memory 3 hours later. But in between, we did imaging experiments in the first hour after the conditioning. And as you can see here, so here you have in blue the flies that have been conditioned, and in gray it's control flies, which also went in the conditioning machines and received electric shock and others, but separated in time. So those flies received the same stimuli as the blue one, but they did not associate them. So this is the closest control that we can design for this experiment. And you can see that the slope of pyruvate accumulation in between those two conditions is different, and basically the learning, the associative learning induces an increase in the pyruvate consumption in mushroom body neurons. And so to go directly to the result, we could show that the pyruvate that is consumed by mitochondria in mushroom body neurons is produced out of the amino acid alanine through an enzyme which is called alanine aminotransferase. And we can, because of the genetic tractability, or thanks to the genetic tractability of Drosophila, we can knock down this ALAT enzyme specifically in mushroom body neurons, in which case we lose this increased pyruvate metabolism. And consistently, the flies where ALAT has been knocked down show, so this is now the memory score measured 3 hours after conditioning. In gray and white, these are genotypic controls, and the flies where ALAT is inhibited in mushroom body neurons show a strong reduction in their memory. So yes, learning induces has an impact on the brain and on the energy metabolism of mushroom body neurons, and this upregulation is critical for memory encoding. So we wanted to complete the description of the metabolic scheme underlying memory formation, and we asked where does this alanine comes from. And then this is, of course, we were inspired here by the mammalian literature about neuron glia metabolic coupling. So we got interested in the implication of glial cells in memory formation, and what we found out is that this ALAT enzyme is also active in cortex glia. So cortex glia is this type of glial cell that I mentioned before, which is enwrapping the neuronal cell bodies. Except that in cortex glia, ALAT works in the opposite way. It converts pyruvate into alanine, and then this alanine can be shuttled to mushroom body neurons. And the way we showed this is that this time we knocked down ALAT in cortex glia, and again we could show that the metabolic regulation induced by learning was lost. So we went further up to the process, and I will not show you all the data. This is an available preprint if you are interested, but basically the global picture is that after a single cycle of training, there is a metabolic upregulation which involves a compartmentalization of glucose oxidation. The partial oxidation through glycolysis occurs in glial cells, and the output of this process is alanine that is transferred to neuron, so that it can be converted back to pyruvate and fuel mitochondria. So this is a nice and clear picture where everything seems very streamlined, and it could be the case if all the metabolized glucose, pyruvate, alanine were plentiful and available with no limit, so that each enzyme could take what it needs in an available reservoir. But actually, this is not what happened, and as we will see now, there are some. So this scheme is critical for memory to be formed, but there are some mechanisms, active mechanisms that ensures that this scheme and not other one that could interfere with memory formation occur. And So this we found because we were also interested in the modulation of glial metabolism, and we happened to find a neuropeptide signaling that acts on cortex glia. So neuropeptides are like hormones. It's signaling molecules that are secreted by some neurons and can, contrary to neurotransmitters, they can flow into the brain to have a global modulatory effects. And this, so we found out basically that the receptor of a particular neuropeptide, which is called diuretic hormone. So when we inhibit this receptor in cortex glia, we see a strong accumulation of lipid droplets. So lipid droplets, we measured them by immunofluorescence using a body PI, which is a specific fluorescent probe that people use to look at lipid droplets, indicating that this neuropeptidergic signaling through this receptor normally inhibits the synthesis of fatty acid in cortex glia. So you can see here so maybe here it's not quite clear, but you see here that we are in cortex glia. So here the black holes would be the neuronal cell bodies, and in between you have cortex glia and those very bright spots indicating fatty acid accumulation. So why is it interesting regarding the memory process that I've described before? It's interesting because fatty acid synthesis involves another enzyme, which is called acetyl-CoA carboxylase, ACC. And through another sequence of reaction, it basically takes some pyruvate and the carbon that ultimately will be accumulated under the form of fatty acid in lipid droplets come from pyruvate. So the activity of ACC can interfere or can compete with ALAT for the available pyruvate pool, providing this pool is limiting. So what we did is in those flies that accumulate fatty acid, we looked at their memory performance. And you can see here that when the receptor is inhibited, indeed those flies are not able to form memory anymore. And when we did the pyruvate imaging experiments, we saw that in mushroom body, in the neurons, those flies were unable to develop the increased pyruvate consumption. So of course, we thought that it was because pyruvate was taken by ACC in cortex glia instead of being available for conversion into alanine. And to test this hypothesis, what we did is a double inhibition of both the receptor and this ACC enzyme, saying that maybe if we prevent ACC from being active, then we will rescue the process. And actually, this is what happened. So here, this yellow bar here is the double knockdown of both diuretic hormone receptor and ACC. And in that case, both the memory is rescued and the pyruvate increase after learning is rescued as well in mushroom body neurons. So the neurons that secrete this peptide are known. So we also looked at the peptide content in these neurons in the brain after learning. And you can see here that actually, so if we look at so this is the red staining here, in the time course after learning, you can see that there is a time window of 1 hour where the peptide content in those neurons is strongly decreased, indicating that indeed learning induces an increased secretion of this peptide so that fatty acid synthesis in glia can be inhibited and let pyruvate available for memory formation. And so beyond the molecular details, why is it interesting? It's interesting, I think, because all this happens in flies that are fed ad libitum. So one could think that, okay, metabolically speaking, they have everything they want. So there shouldn't be any problem of energy availability. But actually, there is. And in order to properly form their memory, those flies need this active neuropeptide-mediated mechanism to prevent the competition for pyruvate and let pyruvate be available for neuronal fueling. So this is, I think, the first message that I want to give today is that the metabolite availability in the brain can be a limiting parameter, even in animals that are in a metabolically favorable situation. Okay, so in the second part now I want to talk about a slightly different question, which is more related to the formation of long-term memory. Because so I've talked only about memory after one cycle here, as I told you that the memory decays after a couple hours. But flies, like many other species, can form and may have benefit in forming a longer-lasting memory. But because long-term memory is very persistent, it needs to be a regulated process, meaning that even for cognitive reasons, it's not efficient to encode long-term memory for any learning experience. First, because it can cause mental overload, and also because it can induce maladaptive strategies. For example, if a fly or if you smell the odor of a banana with something by accident which is negative, and then directly you associate banana with something negative and you avoid banana for the rest of your life, then it's not it's a maladaptive strategy. So it means that there are some inhibitory default inhibition mechanism that prevents long-term memory formation and keep it only for learning experiences that are particularly relevant. So this is a general property of long-term memory, but actually this can be experimentally implemented in Drosophila and studied by inducing memory with different training protocols. Historically, the first attempt to increase the memory persistence was done by simply increasing the number of training cycles, with the idea that more information will induce more persistent memory. And indeed, it works a bit, because you can see that after a masked training cycle, the memory is a bit more persistent than after one cycle. But still, after a couple of days, the memory performance is almost abolished. But if we do a specific pattern of training, which is called the spaced training, same number of cycles as a mass training, but with rest intervals between the training cycles, then flies become available to encode a very stable long-term memory, which can last for several days, up to 1 week, which is very long for a fly, which on average in the wild will live for 2 or 3 weeks maximum. So this spaced training versus mass is a very convenient way experimentally to address this question of the mechanism that will be involved in the triage of the relevance of the information, so that long-term memory formation can be induced only for some highly relevant informations. So, as I said, there is a cognitive reason for filtering the formation of long-term memory. But there is also a metabolic one. And because long-term memory, because of its persistence, because of the genetic reprogramming that it requires, is a very costly brain function. And this, we could measure it directly in in the flies by measuring the food intake after conditioning. So the experiment is what was called in the literature the capillary feeding assay. Single flies are put in a tube where they have access to a capillary filled with water and sucrose. And then the more they drink, of course, the lower will be the level of the fluid in the capillary. So by measuring the height of the water column here, we can measure the sucrose intake by the flies. And we did this experiment after conditioning. And what we observed is that specifically after training, after space training, so when flies form long-term memory, there is a time window of 4 hours where the increase of sucrose is strongly enhanced. The intake, of sucrose is strongly enhanced compared to the controls. And this is occurring in a very early time window of after conditioning. So even before the long-term memory is actually encoded and retrievable. And strikingly, this does not occur after a masked protocol, showing that this is really specific to long-term memory. And to me, this is a direct indication that also, so those flies again have been fed ad libitum before the training. So even in fed animals, these experiments show that long-term memory formation can have a very strong impact on the overall energy budget of the organism. So there are two reasons, both a cognitive and a metabolic one, so that the brain will keep long-term memory formation, yes, only for specific learning experiences.
So
yes?
how many spaced training are in 4 hours? So what's the actual difference between masked and spaced during the 4 hours?
So the protocol that we do is we do the training and then we put them in the capillary. So we measure food intake after the completion of the training. The space training lasts for 1 hour and 30. And this control group here also stay the same duration in the tubes. The mass training, because they don't have the intertrial interval, it lasts only 25 minutes. So of course, it's much shorter. But so I think your idea was do they eat more because they stay longer in the? So I think because of this control, we can be safe that.
So is it really mass versus space or time of being challenged? Like, because it's 25 minutes of something happening versus 1 hour 30 of even if our interstimulus interval is still something quite dynamic, no? So if there was the 1 hour 30 of mass, would it be different?
It may be different because if they receive electric shock continuously for 1 hour and 30, then they will receive much more electric shock and odor than those ones here. So that's why we didn't want to compare directly space versus mass, and we had this specific control for each group, which stayed the same duration in the conditioning machines. So here the blue group stayed 1 hour and 30. Here the blue group stayed 25 minutes. And overall, they all received the same amount, 5 times electric shock and 5 times odor presentation. Yeah. Okay, so yeah. So the question now that to come back to the initial question was how does this filtering occurs? And in 2012 we had found the very important role, critical role of a specific type of the dopaminergic neurons innervating the mushroom body. Those neurons here are called MP1 neurons. And doing calcium imaging experiments, we could show that those neurons show completely different patterns of activity depending on the conditioning protocol that was performed. basically, after space training and for up to 2 hours after the end of the conditioning protocol, those neurons show very strong calcium oscillations, very rhythmic behavior. And as you can see, if we plot a power spectrum, this results in a very nice bell-shaped curve. And on the contrary, after mass training, those neurons tend to be silenced. So those neurons are really, according to our experiments, the master regulator at the right after the end of the conditioning protocol that will determine whether the mushroom body neurons will engage or not into the formation of long-term memory. So this is basically what I've just said. So of course, we wanted then to understand what inside. So those dopaminergic neurons release dopamine, which probably activates a dopamine receptor, and we wanted to understand inside mushroom body neurons how this process of long-term memory gating, so the triggering or not the consolidation of the learning experience into long-term memory was performed. And it happens that we also did some pyruvate imaging experiments after spaced and mass training. And what we showed is that after space training, so when the flies do long-term memory, we also measured an increase in pyruvate consumption, but not after mass training. But compared to one cycle, this increased pyruvate consumption lasted much longer, up to 4 hours after the end of the first training cycle, showing that this increase in this metabolic regulation, the increased duration of this metabolic regulation, could be also important for memory consolidation. So we had these two processes. On the one hand, oscillation in the calcium activity of MP1 dopaminergic neurons, and a prolonged metabolic upregulation in mushroom body neurons that were both important for long-term memory formation and occurred in the same time window. And by the way, it's also the same time window where the fly tend to eat more sugar after space training. So of course, we thought that maybe all those processes are connected. And what we did is an experiment where we expressed in MP1 neurons a thermosensitive ion channel, which is called TRPA1. And when neurons overexpress this channel, and if we raise the temperature, then we can artificially activate those neurons. So this is an experiment that we can do in Drosophila, because Drosophila is an invertebrate, so the temperature is not regulated. And if we put the flies at 30 degrees, then their body becomes at 30 degrees, and if they are at 18, the body temperature comes back at 18. Of course, this would not be doable in a temperature-regulated mammalian body. But here, so what we did is shortly do 2 pulses of activation of MP1 neurons, and immediately after measure the pyruvate consumption in mushroom body neurons. And you can see that here, compared to a control that where we do only we put the flies at high temperature but without expressing the TRPA1 in MP1 neurons, you can see that the activation of those neurons is sufficient in itself to induce an upregulation of pyruvate metabolism in mushroom body neurons. And if in addition we inhibit one of the dopaminergic receptors, which is called DAMB, encoded in the Drosophila genome, we lose the effect. And in behavior experiment, we could show that flies where this particular DAMB dopaminergic receptor has been knocked down, those flies have a long-term memory defect when we measure memory 24 hours after a space training. And consistently also, if we measure pyruvate consumption after space training in flies that are mutant for the DOM receptor, they fail to develop the metabolic upregulation. So these experiments directly connected the activity post-learning of dopaminergic neurons to energy metabolism. And at that stage, we were still left with this scheme. So one direct activation of MP1 DOM onto the activity, the pyruvate consumption of mitochondria, and a role on the gating of long-term memory. So are these two arrows in the end connected and concerning the same process? So recently, these are new data from a PhD student currently in the lab. We got interested in a specific enzyme, which is called PKC delta. We thought about looking at PKC because the DOM receptor is coupled to GQ protein, and the big family of PKC proteins are one of the major class of enzymes that can be activated downstream of GQ pathway. And PKC delta, according to the mammalian literature, especially on mussels, is known to be a regulator of the activity of the pyruvate dehydrogenase complex. And here we could show that on the Drosophila brain, in mushroom body neurons, if we apply PDBU, which is a pharmacological activator of PKC delta, we also are able to induce an increase in pyruvate consumption, which we lose, the data are not here, but which we lose when we knock down PKC delta. And here it's an experiment where we do pyruvate imaging after spaced training with PKC delta being inhibited in mushroom body neurons. And again, we lose the metabolic regulation induced by the conditioning. And of course, consistently with this, when we try to measure the memory of those flies with PKC delta inhibited, we can observe a long-term defect in long-term memory formation. So, what is the mode of activation of the way PKC delta is modulating pyruvate? I told you that we were inspired by the mammalian literature, and one of the hypotheses was that PKC delta is actually an inhibitor of this enzyme PDK. PDK stands for pyruvate dehydrogenase kinase, and it's an enzyme that can phosphorylate the PDH complex to decrease its activity. So PDK is an inhibitor of PDH, and our hypothesis was that maybe PKC delta could inhibit PDK to induce a stimulation of the global activity of the PDH. And we tested this hypothesis by inhibiting both PKC delta and PDK in mushroom body neurons. And as you can see here, we were able to fully restore the formation of long-term memory. So all in all, we end up with this picture of the mechanisms of gating of long-term memory, where MP1 neurons activate the dopaminergic receptor, which in turn induce a PKC delta activation and translocation into mitochondria, which activate the pyruvate dehydrogenase complex through the inhibition of this PDK enzyme. So I think this is very nice because it really bridges the signaling and the information processing domain, because dopamine is like an archetypal neuromodulator involved in many functions, but in particular punishment or reward signaling. And the fact that here through this quite simple signaling cascade, that we can bridge dopaminergic signaling with metabolic regulation really shows that the information and the energy domain in the brain are connected. And so from this scheme, we wondered, in the end, is energy metabolism in itself, or the increase in energy metabolism in itself, gating the formation of long-term memory? And to test this hypothesis, what we did is that we tried to artificially stimulate mitochondrial activity and see the effect on memory dynamics. And so we did those two experiments. So maybe I will start with the one on the right. What we did here is genetically inhibit the famous PDK enzyme in mushroom body neurons. So because it's an inhibitor of PDH, then it will induce a baseline stimulation of mitochondrial metabolism. And as you can see here, in those flies, we are able to increase the memory and produce long-term memory only after 2 space cycles of conditioning instead of 5, which means that by inducing this metabolic stimulation, we are able to facilitate the formation of a long-term memory. And we obtain a similar memory increasing effect by artificially activating the dopaminergic MP1 neurons, which are upstream of PDK. So all in all, those two experiments really, to me, show that in this process of the control of long-term memory, the upregulation of the energy flux is really used as a signal that occurs very early after the end of the conditioning to tell to mushroom body neurons, yes, this is a relevant information. You can enter into the process of making long-term memory for that particular experience. And so this is the second message that I wanted to deliver today, that yes, in addition to information processing, neuronal energy fluxes not only support the activity on neurons on demand, but can be used as instructional signal to control, especially to control a costly process such as long-term memory. Okay, should I stop or? The last part is only 5 minutes, and Natalie has talked about it already, so I will just briefly finish my presentation with so something that you have heard already in the previous talk. This system, this dopamine-based system that controls long-term memory formation, can be used also by the fly brain to implement a strategy, an adaptive strategy to adjust the property of memory formation to the physiological context of the animal. And indeed, we were able to show that when we put the flies in a state of starvation, this will strongly inhibit the activity of MP1 neurons. And after a space training, although in the fed state we have this this bell-shaped curve that I showed you before, betraying the calcium oscillation in those neurons, these oscillations disappear in the starved state, and as a consequence, the formation of long-term memory in those starved flies is inhibited. And when we do, when we force the activity of MP1 neurons, this is the experiment, it's a different part of the data, but this is the experiment that Natalie has shown previously. When we force artificially using the TRPA1 tool that I mentioned before, the activity of MP1 neurons, we can force the formation of long-term memory in those flies, but at the expense of a reduced survival to the starvation regime. So basically, this experiment indicates a trade-off in the fly brain between forming long-term memory, which in the state of starvation may not be very useful, because if you can't find food and you are about to die within the next 24 hours, you may not need to encode a memory that will last for weeks. So a trade-off between this robust LTM formation and the survival to the starvation challenge. But still, so this is another figure from this paper, and so where we compared fed and starved animals after a space training. So this is the regular long-term memory formation, and Kramer is a specific mutant for long-term memory formation. So this indicates that in the fed state, flies form their long-term memory. In the starved state, 24 hours after a training, they are still able to form a memory. But this memory is not sensitive anymore to LTM mutants, showing that it's a different kind of memory with a reduced stability. And recently, just to go, and I will finish with this, we showed that actually this memory that is formed after starvation, which is less robust than LTM, than long-term memory, relies on a completely different metabolic scheme. In that scheme, the neuron and glia system does not rely on glucose anymore, but instead, glia starts to consume their lipid droplet store through a process called beta oxidation, and at the end of this process produce ketone bodies, which are polymers of acetyl-CoA. And these ketone bodies are sent to neurons where they are consumed by their mitochondria to fuel the formation of this semi-consolidated memory, which is less stable than long-term memory. And so this shows that there exists actually a fuel flexibility within the brain, which is capable of producing its own ketone body to fuel a degraded form of memory that may be more adapted to the physiological situation. So this is the last of the three messages that I wanted to tell you today. The existence of a trade-off between that rely on different on fuel flexibility between memory persistence and the optimization of survival. So this is applied to memory. This is, I think, conceptually quite close of what Nathalie has shown in her talk about the trade-off between adaptation to starvation and decrease in the precision of visual coding. The two other messages, I think, are more original, but I'm also quite confident that they will be conserved as well in mammals. And then also maybe, but it's not my expertise, maybe there could be used also as to design or to draw design principles for machines or networks that would be more energy efficient. So to finish, I would like, so I have presented you the work that started in 2012. So it has involved several generations of PhD students and postdocs in the lab. So they are all listed here. Some of them have left the lab for a while. Some of them, and especially Tiffen, Raquel, and Yasmin, are still there. I would like also to acknowledge the work of so Alice Pawlowski, who is assistant professor in the team, and Thomas Prea, who is my co-PI, and we have led basically all those studies together. So I think it's time that I finish. So thank you for your attention.
Further questions?
Maybe I missed it, but what's the effect when you block the dopamine? You test it on the space memory, but what's happening with the mass memory?
On this particular neuron, nothing. It has no effect on the mass memory. No. It does not. So when we block those neurons after space training, we lose the ability to form long-term memory. But after mass training, there is no effect. So it's really specific to.
Yeah. I'm trying to figure out when, at what point of the protocol, there is a decision made. So you show there's the training, there's the same training, so it must be temporary at some point.
I think the timing, and that's why it's interesting that all of this occurs in the first hours, because when we measure the memory, we look 24 hours after the conditioning protocol. But all this regulation that I've described occur within, yeah, I would say 3 hours after the conditioning. So it's even before any type of signaling that can go through the cell nuclei and trigger genetic reprogramming. So that's why we think it's really a gating mechanism.
So then would be the amount of dopamine, because these MP1 neurons would be more active, is it that?
They are more active. This we see it with the calcium imaging experiments. So of course we could use, now there are dopamine, so we did calcium imaging at that time. Now there are dopamine sensors, so we could use them to directly see if indeed there is more dopamine released after space than after mass. But yeah, this we have not formally checked.
But that's the idea.
Yeah, that's the idea. Yeah. Yeah.
But do you expect that during this space protocol, during the interval,
Yes.
these dopamine neurons are active actually?
Yes.
We see them after that, but
Yeah, we see them after because we don't manage yet to do the conditioning directly under the microscope. Ideally, what we would like to do is do every image during the whole process and see, all along the space training protocol if the activity is progressively increasing and with which timing. So, but yeah, this is the idea. But the more cycles we do, providing that there is the rest interval, the more cycles we do, the higher, the more consistent the oscillatory behavior will be.
Other questions?
Do you expect it will be different if the spacing between the training period is irregular?
So we have not done imaging on that, but we have done behavior experiments, trying to do random duration of ITI, and this is the same. This is not less or more efficient, but so just in terms of on the memory performance, it's similar.
Okay, so last question.
No, because if I understood correctly, if you force a starved flies to memorize, they dies early. Why is the mechanism? Why this flies dies early?
They die early because they are on starvation. We measure the time of death in the context where they are deprived from nutritional input. They die earlier because we force a process to occur that is very costly metabolically, and I think we should put this result in parallel to this one. When they have food available and they do long-term memory, they eat more. When we force long-term memory and they cannot, so something that I didn't say is that they eat more, but this is not required for memory formation. If we put them on starvation during the 4 hours, only during the 4 hours, they will form long-term memory normally. This is no problem. So this is just a consequence of the fact that the machinery underlying long-term memory is costly. But still, it betrays the metabolic cost of long-term memory. If they are now in a context where they have no food access and where normally long-term memory is inhibited, and we force long-term memory to occur, then we force an expense that was not normally planned. And because long-term memory has a strong impact on the energy budget, then in the end, they will die earlier.
Okay, so thank you very much, Philippe.
Thank you.